Clinical chemistry, hemoglobin and bilirubin. The role of hemoglobin is to transport oxygen to the tissues and, to a much lesser extent, transport carbon dioxide to the lungs. This is a picture of hemoglobin here. Hemoglobin consists of 4 globin proteins, 4 main subunits. At the center of each one of those globin proteins is a heme group, and at the center of the heme group is the iron molecule. Iron is able to bond to oxygen, and that's how oxygen is transported through the blood. Hemoglobinopathies are diseases that are related to defects in hemoglobin structure. So talking more about the protein components, there's those 4 subunits that you would see. And depending on what type of hemoglobin we're referring to, there's going to be different types of subunits. Embryonic hemoglobin comes in a Gower1, Gower2, and Portland version. The Gower1, for example, has 2 zeta chains and 2 epsilon chains. Fetal hemoglobin comes in 2 alpha chains and 2 gamma chains. Adult mammalian hemoglobin is 2 alpha chains and 2 beta chains for the most part. But in 1 to 3% of the hemoglobin molecules in the human body, you will see hemoglobin A2, and that's 2 alphas and 2 deltas. The genes for hemoglobin expression are chromosome 16 and chromosome 11, and you can see which genes are on which chromosome. Hemoglobin exists subunit cooperativity. What this means is that when oxygen binds to one of the subunits of hemoglobin, it causes a conformational change in that globin protein, which then affects the neighboring globin protein. And it allows for oxygen to bind at that neighboring globin protein at a higher affinity. Once that second one is bound, it causes another conformational change, which allows oxygen to bind to the 3rd subunit at a higher affinity. And it does so again for the 4th subunit. So that when one binds, it's kind of a chain reaction that increases the affinity of oxygen binding in that molecule. The reverse happens when oxygen is released. Fetal hemoglobin is the predominant hemoglobin in the newborn. It's normally absent in adults unless there's a pathological condition. So there's a genetic component where you could have beta or delta beta thalassemia, the hereditary persistence of hemoglobin F, or sickle cell anemia. And then there's acquired. So it can be acquired midterm pregnancy from bone marrow suppression, leukemia, thyrotoxicosis, or hepatoma. Something to make note of with fetal hemoglobin is that babies do not get malaria. And the reason for that is malaria, the Plasmodium parasite, likes to eat hemoglobin, and it's not too capable of eating fetal hemoglobin. When the baby is about 6 months of age, they have a big shift in the amount of hemoglobin that they have, so their fetal hemoglobin gets really low and their adult hemoglobin goes really high. So the majority of the hemoglobin is hemoglobin A at 6 months of age. And at 6 months is when you start to see kids getting infected with Plasmodium or with malaria. So that was fetal hemoglobin. Now we're going to talk about some of the other hemoglobins and the pathological conditions. So hemoglobin S causes an amino acid deficit at the 6th position on the beta chain. That detail is important not only for the upcoming test, but also for the qualifying exam. You will be asked questions on what position on what chain is this defect. So you should know hemoglobin S is an amino acid defect at the 6th position on the beta chain. It is the most common hemoglobinopathy in the United States. Individuals have the sickle cell trait or sickle cell disease, and this is more prevalent in Black Africans and African Americans. Hemoglobin C, the glutamic acid in the 6th position of the beta chain, is replaced by lysine, resulting in a net positive charge. This is found in West Africa in 17 to 28% of the population and 2 to 3% of African Americans. Hemoglobin SC is the most common mixed hemoglobinopathy. The beta genes code for beta S chains and beta C chains, leaving no normal beta chains to produce hemoglobin A. Hemoglobin E, an amino acid substitution of lysine for glutamic acid in the 26th position of the beta chain, results in a net positive charge. This predominates in Asia. Hemoglobin D is a variant with electrophoretic mobility on cellulose acetate, like that of hemoglobin S, but with a negative solubility test. And then there's quantitative defects, the thalassemias. Thalassemias are a group of diseases in which the defect causes reduced synthesis of one or more of the hemoglobin chains. So alpha thalassemias cause a reduced synthesis of the alpha chains. Beta thalassemias causes reduced synthesis of the beta chains. Solubility test is a screening test for sickling hemoglobins. So sickling hemoglobin typically precipitates in solution. So that's the solubility test is based on that principle. And then there's two different types of electrophoresis that can be performed. There's the cellulose acetate and the citrate agar electrophoresis. Cellulose acetate occurs at a much higher pH, so an alkaline pH. Citrate is going to be performed at a lower pH, an acidic pH. This is a picture of what that would look like. This right here is the origin, and then the hemoglobin molecules or subunits, proteins, are going to migrate through at this rate. You should be familiar with which one migrates the furthest, the second furthest, and the migration pattern on here. Again, not only for this class, but also for the qualifying exam. And it's not just for the cellulose acetate, you should also pay attention to migration patterns on the citrate agar. So here you can see on cellulose, hemoglobin A is going to migrate the furthest. Citrate agar, the origin is actually in the middle. And depending on the charge of the protein at that pH, it's going to migrate either toward the positive if the protein has a negative charge, or it's going to migrate toward the negative if the protein has a positive charge. So you can look at the migration patterns and where it ends up on the citrate agar. Hemoglobin A2 quantitation is best accomplished by microcolumn chromatography or high-performance liquid chromatography. There's the acid elution stain for hemoglobin F. It distinguishes erythrocytes that contain increased amounts of hemoglobin F from normal adult cells. And then hemoglobin F quantitation is based on the principle that fetal hemoglobin is resistant to alkali denaturation in 1.25 moles per liter of sodium hydroxide for 2 minutes. There's also DNA technology. The definitive diagnosis of some hemoglobinopathies and thalassemias that involve combinations of genetic defects may require DNA analysis. DNA sequence of interest may be easily analyzed from whole blood or spots of dried blood on filter paper. The good thing is that it is definitive and it gives you a lot of information, but the bad thing is that it is at a higher cost and not many labs have this capability. It's especially useful in prenatal diagnosis of thalassemia major and sickle cell anemia. So talking more about the synthesis and degradation of hemoglobin. So hemoglobin is made in immature red blood cells in the bone marrow, 65% nucleated cells and 35% reticulocytes. Normal synthesis depends on adequate iron supply and heme protein synthesis to form the globin portion. So if you don't have enough iron in the diet, you're not going to be able to produce enough hemoglobin. Degradation can happen in two different pathways. There's extravascular or intravascular. Extra means outside of, vascular is referring to the blood vessels. So extravascular is going to occur outside of circulatory system, and that typically happens within the phagocytic cells of the spleen, liver, and bone marrow. Intravascular, intra means inside of, is released directly into the bloodstream and dissociated into alpha and beta dimers. When hemoglobin is degraded, it's broken down into bilirubin. Bilirubin is a waste product, and this waste product has to be eliminated from the body. If bilirubin accumulates in the body, it can cause a whole bunch of issues, including brain disturbances and even death. To manage or to form hemoglobin, we start off with the heme group. So bilirubin, we start off with the hemoglobin. So hemoglobin is broken down into its subunits, and the heme group is cleaved. And when that heme group is cleaved open, it's converted to biliverdin. Biliverdin is then cleaved again to produce bilirubin. Biliverdin is green in color, bilirubin is yellow in color. Unconjugated bilirubin is the waste product of hemoglobin breakdown. So when we break down hemoglobin, that heme protein turns to biliverdin, then bilirubin, that's unconjugated. That then has to be transported to the liver to be conjugated. Conjugated bilirubin is water soluble and excreted into the bile and eventually the GI tract for elimination. So when we break down red blood cells, the macrophages are going to break down as much as they can, as possible, break down and release those heme groups. Heme is going to be transported to the liver bound to albumin, and then it's released into the liver, enters into the hepatocytes, where it's going to be conjugated. Bilirubin is secreted by hepatocytes in the biliary canaliculi, and then converted to urobilinogens by bacteria in the gut, and eventually excreted in feces. So here you can see it's broken down here. It's converted into bilirubin. This is unconjugated bilirubin. Unconjugated bilirubin is going to be bound to albumin, and it's going to be transported to the liver. Once it's in the liver, it's going to be conjugated. Conjugated bilirubin is going to be excreted into the digestive tract through bile, and then it's going to be converted to urobilinogen and excreted in the feces. It can also be excreted in the urine. Alpha bilirubin is unconjugated, beta is monoconjugated, gamma is diconjugated, and delta is irreversibly bound to albumin. It's measured in serum and urine by a variety of techniques: chromatography, electrophoresis, photometric, and chemical methods, and there's also the Diazo reaction. Diazotized sulfolinic acid, the Diazo reagent, reacts with bilirubin to produce 2-azodipyroles. It's reddish purple at neutral pH, but it's blue at low or high pH. Direct bilirubin is conjugated bilirubin plus the delta bilirubin. Total bilirubin includes bilirubin plus the direct bilirubin plus the indirect bilirubin. Indirect bilirubin is unconjugated. The way that we get unconjugated bilirubin to be detected by the total bilirubin test is by adding an accelerator. Accelerators include methanol or caffeine, and there's a particular procedure, protocol that you would follow in order to add that accelerator. So the accelerator is going to allow the unconjugated bilirubin to react with the reagent, and then you're going to get a total count of both direct and indirect bilirubin. In other words, conjugated and unconjugated bilirubin. In order to measure unconjugated bilirubin, then you would take total bilirubin minus the direct bilirubin, and whatever's left would be the unconjugated bilirubin. Specimen collection, you want to avoid hemolysis and lipemia. Typically, a morning specimen is better. Avoid icteric samples, protect from light, and then refrigerate. It's stable for 3 days at 4 degrees Celsius and 3 months at -70.